Hello. Hi. Can you guys hear me? Okay. Hello, everyone. I'm Malishi Joshi, or Molly. I run the functional genomics core here on this campus, and it is my privilege to introduce today's guest, Ralph Garippa. Did I say that right? Okay. I've had the privilege of knowing Ralph for a while. I consider him a part of my professional tribe. He has, as I navigate the complexity of running a core facility on our campus, he's always been very gracious with offering scientific and administrative assistance, and his guidance has been very invaluable to me. So Ralph's scientific journey has been extraordinary, from his undergraduate work to his PhD at Columbia University. He's always pushed the limit, and um, after earning his doctorate, he moved over to Roche, and he has had a very successful career at Roche for over a decade. He was instrumental in establishing their high content screening platform. And then he was also instrumental in combining that with RNAi technology, which has led to identification of several novel targets for oncology. He's also one of those rare people who's had a very successful career in industry and has successfully transitioned back to academia. Currently, he is the director of the gene editing and screening core at Memorial Sloan Kettering, where he has been instrumental in developing the facility and offering cutting edge technology to the researchers on that campus. And his numerous publications are a testament to the amazing work that he has done. And he's asked me to keep this short and sweet and let his work talk for itself. So without further ado, I would like to welcome Ralph Garippa. And before we go on to his seminar, I would like to give him this Cancer Warrior Award as a token of appreciation from our Cancer Center. Okay, Ralph, it's all yours.
Thank you. Well, thank you, everyone, for coming to the talk today. So, as Molly said, I run a genomics screening core at Memorial Sloan Kettering in New York City. Very similar to what Molly does for you. I've been doing it a little bit longer. And so that means I have built up at least a few stories that I could tell. And so I thought today I'd really give you a sense of how a core facility can enable a top flight site like you have here. And also, I picked these 4 stories for a very specific reason, and that is that they were all led by a graduate student who eventually published, and it became the centerpiece of their thesis defense. And each one of these 4 projects still has a life, a very good life in research. So with that, let me move on and just start with my disclosures, which are here. It's a very short list. And this is my core. So I have 6 full-time scientists in the lab working. This is Jessie here. She's in charge of our pooled screen. This is Claire. She's in charge of all of our imaging platforms. Sanjay and Hannah are our molecular biologists. They do all of our cloning. This is Annie Magpusau. She's in charge of our chemical library, all of our high throughput screening. John Min Yang is our lab manager. He does all of our virus production. And then here, hiding very pregnant in the back, is my admin, Anna Crawford. So Anna's actually a graduate of University of Colorado at Denver. She did her master's degree here, and she looks out for me. So whenever I go on the road, she has, just seminal words of wisdom. So her best advice for me, she said, if you put up this next slide, everything will be fine afterwards. So go Buffs, and let's see how the rest of this goes. So I'm only going to spend one slide on my core facility. And the reason is I want the work that we do to speak for who we are and what we do. So there's 29 core facilities at Memorial Sloan Kettering, everything from animal imaging, antibody development, in vivo anti-tumor assessment, cell therapy, including CAR T, flow fax. Here's our humble core, gene editing and screening. We really started out as the RNAi core. And then when CRISPR came along, we quickly converted our pipeline to making CRISPR. And then we also brought in the high throughput screening and high content, so they renamed us, and that's our new name. So these are all the cores for some very sophisticated work that's being done, stem cell research down to media prep and glass washing. So that's our cores. All right, so the first story I'd like to tell you is about a collaborative project that we did with Daphna Barsaghi at NYU. So this was a high content, full genome siRNA screen. So every one of the wells had 4 siRNA dedicated to a particular gene target. We did it this way because the imaging, the phenotype of the cells was key, and we couldn't get that data any other way. So let me explain. So this is one of our high content screening systems, the InCell 6000. It's a laser line scanning confocal system. We also have the Yokogawa CV8000, which is state of the art. But this is a really good system. We can do Z-stacks for spheroids. We can do 2D, lipid droplet. And we, because we're a core facility, we either reserve the time for ourselves to do internal projects, or we make it out available in hourly or daily rates. So the principle of the screen that we ran is exemplified by the screen I ran a number of years ago, looking at 7 transmembrane receptors. So very quickly on the biology, you have a GPCR at the cell surface. It binds an agonist. It undergoes a conformational change, and then it attracts a G protein-related kinase to phosphorylate the cytoplasmic domain. And then the reporter in this system is beta-arrestin, which is recruited to the activated receptor, and that's labeled with GFP. So these activated receptors are then pinched off into clathrin-coated pits, and then they go through a rapid or a slow recycling. What that looks like under the microscope is here the diffuse GFP throughout the cell. Here's the clathrin-coated pits, and here's the vesicles. Now, if you put this into a plate reader, you'll get exactly the same intensity reading on all 3. Your eye can pick it apart very quickly, but a plate reader can't. And that's why you need a masking technique in high content. So what is our masking? So for this, we used a DRAC5 red dye that very nicely labels the nucleus. And then you get a little bit of spurious staining outside of that that will define the plasma membrane border. And then you set up a search zone in between the nucleus and the cytoplasmic border for the spots. And you can look for spots that will mean the clathrin-coated pits or the internal vesicles. So that's how we do the quantification for a phenotypic screen. So the screen that Daphna was interested in is studying the process, the rare feeding process that a lot of RAS cancers use, which is macropinocytosis, which is the engulfing of material outside the cell. So macropinocytosis, it's an endocytic process. It allows cancer cells that are driven by RAS to engulf large amounts of material, extracellular, to feed their tremendous metabolic demand. So once the material comes inside, it undergoes proteolytic cleavage. You get glutamine, amino acids, and it feeds several pathways of carbon metabolism. Now, there was some previous work done about 20 years ago where they looked at 2 compounds. One was Vaquinol and another one was Momip. And what this did is it caused the RAS cancer cells to overeat, overdrink. And that process they called methwosis. It's an old Greek word, which means eat to your death. Macropinocytosis, you may or may not have heard of it before. Clathrin, certainly. Phagocytosis, certainly. But there's a lot of parts of it that are misunderstood. So we thought, well, let's see if we can actually make this into a druggable target for RAS. So as I explained earlier, macropinocytosis engulfs particles through ruffling. So our reporter for this cell was HeLa. HeLa actually has wild-type RAS, but very low macropinocytosis. So we created a reporter system where we had DOX-inducible mutant HRAS. So we could turn on and turn off the macropinocytosis so that we'd be able to detect agents that would either increase it or decrease it. So this is what the images look like. You can see the negative control here. This is a high resolution blow up. So you see the engulfment. This is tetramethyl rhodamine dextran. So it's a high molecular weight protein, very similar to what macropinocytotic cells will eat in vivo. And we would leave that on the cells for 30 minutes. We'd wash extensively because it's a dye. You want to get rid of every bit that's not engulfed. And then we would quantify it using the software on the imager. So we decided to use a very short time course so that even though it's a large molecule, we wouldn't have other endocytic pathways contributing. And because it's so bright, we're able to use a very low concentration, saving a lot of money. We set up a plate washer automated so we could do a 6X, which is a lot of washing, and not lose the cells using a side sprayer. And then a Corning cell bind plate really kept the cells adherent for the entire length of the assay. So the assay, the siRNA collection that we used, it's the former Dharmacon collection that was then bought out by Horizon. So 4 shRNA in every well to every gene in the human genome. So we ran that screen and then we looked at the data. So here's the data. So over here on the far left, these are the dead cells. And down here at the bottom, these are the DOX-inducible macropinocytosis. So that's the maximum. The green cloud is the entire collection. Blue here is our negative control. And the red is our positive control. So this is actually RAC1 siRNA, which we know will inhibit macropinocytosis. On this side, we looked at the spot area. And on this side, we looked at the spot intensity. So either one of them could have been used as the readout, but because the Z prime was better, we had better separation here of the negative and positive control. We decided to use spot intensity, which was, in hindsight, a good decision. So for interpreting our data, we used this freeware that's on the web, 9, which is really useful. We ran our screen. We did the data collection. Here's the data visualization. Selected hits, and the hits were either 3 standard deviations above or below the mean. So those were the gene targets that would either enrich or put a damper on macropinocytosis. So a lot of the hits that came through, we expected. We expected RAS family members. We expected transport proteins that came from the RAB family. Altogether, we had 960 hits out of 20,000 genes with RAS and RAB showing up, but importantly, a number of novel genes and gene families came up, which really gave us great insight into macropinocytosis. Now, the graduate student that was doing this was presenting this data to his thesis committee, and one of the committee members said, you know, if this high content screening really is worth its money, the position of the macropinosomes in the cell should give you some indication of what targets are involved. So sure enough, the RAC and RAS related genes had a position very close, perinuclear, but the other gene families that were newly discovered actually had other positions within the cell. So it was informative to look at the very discrete details of the phenotype. So at the end of this, we published our paper. The student graduated with his PhD. This is the paper that we put out with Daphna. And then underneath the double dotted line, that student now is the CEO of the spin-out company, TezCat, that's responsible. So TezCat, their approach now is to take those mechanisms that they know for overfeeding and to put a warhead on there. And so to boost up the macropinocytosis, encourage the RAS cancer to engulf as much of that toxic material as possible. When it starts to dampen with resistance, that feeding, then come in with the inhibitor of the macropinocytosis. So it's a feed, overfeed, and starve, overfeed and starve technology. And it seems to work pretty well. So this is the in vivo data. On this side, you'll see the data with multiple myeloma. And then on this side is with very difficult KRAS. And if you look at the highest dose here, 5 mg per kg of the TEZCAT 102 compound, you see it's almost flat line in terms of tumor volume size. So we're very encouraged by this result from an arrayed siRNA screen using imaging as a readout to be able to get us to the point of really considering this as a possible therapeutic for very difficult RAS-driven cancers. So with that, that's the end of the first story that I wanted to tell you. And I'll look at my watch and be conscious of the time. The second story that I want to tell you will go from RNAi into CRISPR, and actually with a pooled CRISPR screen. And so you all read this at the end of last year. This is Beata Halase. She is a virologist who unfortunately contracted breast cancer, and the breast cancer was resisting conventional therapy. So she took matters into her own hands, and she actually created viruses in the lab to treat her own cancer. And the cancer that she had, the breast cancer, it started to shrink, it became softer, and it became very removable for the surgeon. And she's still into remission to this day. So you could see the virus. This is a measles-based virus that's attacking the cancer cell up here. But there are many other oncogenic viruses that are in clinical trials. And I have a couple of them listed here. The ones that are shown, and this table, by the way, is many pages. I'm just showing you here adenovirus and vaccinia virus, but there are many others. She used measles virus. The one that I'll show you is even a different virus. But you see there's many companies here. Onyx was one of the original companies in this space. There's several others here. Phase 1 trials, phase 2, it starts to thin out when you get into phase 3. But all kinds of cancers, melanoma to pancreatic to brain cancers. So there's a lot of possibility with oncolytic virus. There's only one FDA-approved drug, and that's Imlygic. That's T-VEC. It was approved in 2015. And so this remains to this day, 10 years later, the only approved oncogenic virus. And I think there are some reasons. These are tricky therapeutics to develop. But down here, I have 3 other ones that are now in late-stage trials and fast track, and hopefully will hit the market. But none of them are the ones that we're interested in. The one we're interested in is next. So I went the wrong way, and that's okay. Seneca Valley virus. Seneca Valley virus is a virus that's oncolytic, and it has a high degree of tropism for neuroendocrine tumors. So small cell lung cancer, and as well as these pediatric cancers. This is a big problem. About 30,000 people die from non-small cell or pediatric cancers every year. This virus homes in on those cancers, not all of them, but very many of them. So it's a picornavirus, it's a RNA-based virus, and it's a icosahedral capsid type. I'll give you a little more information on Seneca Valley virus. So we know it binds to a receptor here on the cell surface. It enters, it uncoats, and like many other viruses, it hijacks the transcription translational machinery of the host cell. In this case, would be a neuroendocrine cancer. And so the protein is produced, it's repackaged, and then at some point, hours later, the cell bursts open, and those viral particles go looking for other cancer cells to reinfect. And that's how it really dissolves, liquefies the tumor. What has never been known is the exact receptor. So it doesn't bind indiscriminately onto the cell surface. There are certain receptors that it looks for, but no one knew what that receptor was. And that creates a clinical problem, right? So here's all the drug features of Seneca Valley virus. You can give it IV, and it does not integrate with the host genome. It's replication competent, and it persists in a patient for about a month. It has no DNA stage. It's immunologically stable, and it is not activated by blood components. It does not cause hemagglutination. It homes to the tumor with that single IV injection, and pre-existing antibodies are very, very low, very rare. And the fact that always blows my mind is that it has a greater than 10,000-fold higher potency for neuroendocrine and pediatric cancers than for normal cells in the human body. And it's not known to cause any disease or animals. So these are all good characteristics of a drug. And in fact, when Charlie Rudin and John Poirier did their first in vivo studies, they found some very nice tumor shrinkage by administering. And when they went to high dose virus, actually it just wiped out the tumors. And they created a GFP so that when they injected human cancers into the neuroendocrine, into the hind flank of nude mice, of immunocompromised mice, they were able to visualize the SVV GFP, homing in onto that cancer and leaving other areas of the mouse alone. So that all looks good. And then they went into a phase 1 clinical trial. This is when Charlie was at Johns Hopkins. And you see here's the injection of the virus and the virus load here. About a week later, you start to see neutralizing antibodies. And even though this was a phase 1 study, looking across several patients, in the blue, you see patients who had good response, tumor shrinkage, meeting biomarkers. So, but it's not every patient, right? Some of them had no response at all. This patient unfortunately passed away during the trial. So what's the difference? Why is it working so well in certain patients and not the others? Well, we don't have biomarker. We don't know why. We don't know what that receptor is. So maybe if we could find the receptor, we could start to stratify the patients and create a criteria for recruiting only those patients into the trial. So it wasn't for lack of trying, they looked. Okay, so all of the previous efforts just did not find that surface receptor. They tried RNAi screens with hairpins and siRNA. They tried cDNA libraries. They tried glycopeptide capture. Maybe the receptor protein's too low. Maybe the knockdown is incomplete, right? Because you only need a couple of receptors on the surface for that virus to get in, and then all hell breaks loose. So maybe the knockdown was incomplete. Maybe it's multiple receptors or receptors complexed to proteins that are recruited. So for whatever reason it was, they could not find the receptor. So Charlie and John came to our lab and said, we want to try this new CRISPR technology because it's much stronger, we understand, than RNAi. So we set up the screen at that time. The penultimate full genome library was called Gecko. This came from the Broad. And as you know, Cas9 is the functional unit that will cause the cleavage. When you go down non-homologous end joining, in indel, there's usually an indel, a mutation. That's what you want for the knockout. The other side of that is the more rare event, which is homology-directed recovery and repair, and that we use for cell line development, but not here. So the library itself, it's to just about 20,000 genes. We had, in this case, 3 in library A, 3 guides in library B, and a number of negative controls, so we moved ahead. Now, John and Lindy, the graduate student that was running the project, they made a very important decision. They said, we're going to go after this with 2 different cell lines. One cell line is going to have very high permissivity, high tropism for the virus, right? So you need very little virus to be able to get in and infect. Then we're going to take a second cell line that's on the other end of the spectrum, and as far as we know, it's very hard to infect, but it is infectable. So that one that's lowly infectable became the HAP1. The one that was highly affected by permissivity and tropism is over here on the left, and that's the H446. So we ran parallel screens in 2 very different cell lines that had high and low susceptibility to the virus. And so we optimized the screen for each one. And there were some conditions that were the same and some that were different. You can see those here on the bottom. So the number of cells about equivalent, the MOI for the virus, the same at 0.4, puro selection slightly different. The length of puro selection, that's for the guide library. So you have a stable Cas9 in the cell, and then you bring the puro. So the Cas9 will be under blast or high grow, and then you come in with a puro selectable library so you can get rid of all of your cells that are not carrying a guide with puromycin. And then after you do all that and you take your time 0, you throw in the virus and you let go get the cells. So every cell will die, will burst from virus unless you knocked out that receptor. Okay, so it's the last man standing, it's a positive selection screen, which is the easiest type of CRISPR screen to do. And lo and behold, here are the results for the HAP1 and the H446. So it was unmistakable, right? So now all 3 or all 6 of the guides for these targets showed up outside. So here is the rank and file of all of the inactive ones. These are the cells that got blown open by virus. But look at these targets up here that kept clean. So it's either ANTXR1, TEX2 came through, and then there's a number of other ones that came through as hit, but really those proved to be passenger genes that were just being dragged along. The real action was with the ANTXR1. So now we took the individual guides from the high throughput screen and we tested them individually. And sure enough, it was unmistakable that the ANTXR1 and not the TEX2 was the protein that was going to be the item of interest. So what is ANTXR1? You may know it as TEM8. So originally it had been identified as tumor endothelial marker 8. A protein that's overexpressed in a lot of cancers and endothelial cells. It's a single transmembrane receptor. And the reason it's named ANTXR1 is because opportunistically, anthrax bacillus toxin binds to that receptor. So in some quirk of convergent evolution, anthrax and Seneca Valley virus decided they liked the same receptor for getting into the cell. Otherwise, there's no commonality among the two. So ANTXR1 is actually only one of 2. So anthrax can get in via 1 or 2, but Seneca Valley virus only uses 1, and I'll show you that data. So we looked at a number of different indels, and you see them here in this box. So we looked at whether it was deleted and out of frame or inserted and out of frame. It didn't matter. It was a knockout that was non-functional for binding to the virus. And in fact, all of those cells from all these different cancers were 100% viable. Whereas if we didn't knock it out, that in fact, all of those cells died. And so then we were looking at various other cancers, the same result. So we looked at SCLCs, we looked at HAP1s, Ewing sarcoma. It was the same story again and again. Knock out ANTXR1 and the cells will survive. Anything less than that, and they get blown open by the virus. So in order to prepare this piece of work for publication, John and Lindy, they prepared a GFP that you saw earlier, Seneca Valley virus, and we re-expressed the ANTXR protein in the cells that had been knocked out and were not binding. And in fact, we're able to get virus binding in all of the cases and the ability to knock down the cancer. And this is an HA tag that was also put on the receptor so we could track it. So we thought there must be something else going on. This is a great answer to have the ANTXR1 or TMB8 receptor, but it seemed like there was something else going on. So John and Lindy consulted the Cancer Cell Line Encyclopedia. And in fact, if you look at it in the permissive cells up here, ANTXR, and the non-permissive, what we found is an absolute requirement in permissivity is to have that ANTXR1. But looking across 1,037 cancer lines in the CCLE, they also found a high preponderance here of interferon genes being down. So you need the 2. If you're going to really stratify a patient population, you have to make sure they have ANTXR1 on the cell surface, and they have to have a dampened interferon-based immune response. If you have those 2 things, those patients should look like the ones that were the blue bars in the phase 1 study that I showed you earlier. And then just to wrap this up, they made an FC chimera. And with that FC chimera, you could see when we added that back to cells that had no receptor, that the SVV binding correlated with the amount that we added back. It was not displaceable by high-dose sodium chloride. And the effect is absolutely specific to the ANTXR1 and not to the ANTXR2. So we pretty much felt very confident at this point that we had really found the receptor, found the intracellular conditions with dampened immune response, and would be able to move this. And so now we're starting to see a renaissance in the interest, not only in Seneca Valley virus, but also in cells which express TEM8. And if you look at this, there's at least 7 types of cells that have TEM8 receptors on them in a heterogeneous neuroendocrine tumor. So a great possibility, number one, to use this as a single modality, but number two, and probably more importantly in the year 2025, we have the possibility of taking cold cancers and making them hot and opening up avenues for cell therapy that wouldn't be available before. So even if you did get a neutralizing antibody, you would, in theory, be able to heat up that cancer enough so that you get good targeting with a CAR T or a natural killer cell as a therapeutic. And this is just 3 titles that I pulled from recent literature, where people are talking this up again about oncolytic viruses and the homing capacity, TEM8 as a good biomarker, and then using TEM8 ANTXR1 in a cancer-based therapeutic as Seneca Valley virus. All right, that's the second story. I'll have 2 more, hopefully, that I could tell you about. This third one, I'm going to move away from cancer biology into stem cell biology, but we're going to stick with the full genome CRISPR library, pooled, and show you the power of that. So this is one of my collaborators at Sloan, Danwei Huang Fu. She came through Doug Melton's lab at Harvard. So they study pancreas, they study insulin secretion, they study diabetes. This is Danwei's lab at the time, and this young man up here, Ching Li, is the graduate student who ran that screen. So they're very interested in developing artificial pancreas, right? So an artificial pancreas, if you had a way of really with high efficiency taking a patient's cells and bringing them down endoderm, definitive endoderm into beta pancreatic cells, you have a chance actually to put that into a semi-permeable membrane, either in the arm or in the leg, and it will act like a peripheral pancreas, right? So it will respond to postprandial glucose. You'll get insulin secretion appropriately. The glucose level goes down, the insulin level goes down. So a very attractive way to treat a type 1 diabetic rather than daily meal stimulated insulin injections. So we know a lot about how to make beta pancreatic cells. So it starts with definitive endoderm with a SOX17 label. That then goes to progenitor pancreatic cells, endocrine progenitors, and finally beta-like cells. And there's a number of companies that are looking at this. And as I mentioned, you can go through this in cell culture in a GLP facility. And at the end of the line, you have these pancreatic cells that can go into a semi-permeable membrane that you can implant in the patient, and they'll be free of insulin. But it's not a short process, right? So you need to 16 days, almost a month, right, to go through all of these different stages to get through cells. And time is money, so you can imagine how much money this comes to just to load one artificial pancreas with iPS cells. So Danwei came to our core lab and she said, we know a lot about definitive endoderm differentiation, but with this new CRISPR tool, maybe we could find new regulators, new positive and negative regulators of definitive endoderm and pancreatic cell. So she came to us, and here's the protocol that they use. So when you take the iPS and you add this small molecule inhibitor, it's a chiron compound that will inhibit GSK beta 3. And then you take activin A. So in a very specific fashion, you have to have a short burst with the small molecule and then 3 days worth of the activin A. And when you do that, you get over 3/4 of the cells will produce definitive endoderm, the first journey onto making pancreatic cells. If you mess that up, if you leave out the activin A, it doesn't work. If you leave out the chiron one day, it doesn't work. If you keep the chiron on too long, it doesn't work. So it's a very specific time-related process to get just through that first stage of differentiation into definitive endoderm. So what Donway did, and because stem cells in particular don't like being stably transfected with a bacterial endonuclease Cas9, she made an inducible system, and it also was with a SOC17 GFP readout, so we'd be able to see the movement from IPS into definitive endoderm, that first stage. So she set up the reporter, and then we took the state-of-the-art full genome CRISPR library that you just saw in the Seneca Valley virus, Gecko2, and we set up the screen. So here is the 1000x coverage that we did. We did a high representation because we're looking for dropouts as well as positive regulators. Here's the dox and puro, and then the small molecule plus the activin A. And then we sorted on facts to be able to look for the GFP positive and negative cells. So in pooled screening, there's three main types of readout, right? So here are the plasmids for either SH or SG. You have to package those into a lentivirus. You then infect the lentivirus in your cells of interest. In this case, it's IPS. And then the previous screen showed you dropout, right? The cells that survived by the fact that you knocked out the TMB8 receptor. This screen is fact-based. So now you're sorting for the SOC17 GFP or cells that are not to look for the differentiation. So this is the result of the full genome screen. This is a lot of work. It's 100 cell flasks of 150 millimeter each, fills up half of an incubator, but we were very pleased with the result because a number of the factors that are involved in endoderm differentiation came up. So EOMAS, SOC17, SMAD2, FOXH1, this is a very good sign. But what about the original premise? Can we find genes we didn't know about? And so here is in our MAGIC program is what we do to look for gene enrichment for the dropouts and the ones that are positive regulators. So we came up with 160 possible genes as positive regulators, 287 as negative, and that's either 3 or 2 of the hits from that library in the pool. All right, so we looked at those, and then we did a Z-score and a robust ranking to get the most interesting ones, and then we validated them based on the ones that were the hits. So it turns out that looking at 22 of the top 50, that 16 of those 22 validated, 9. Now I want you to think about this with all of the stem cell work going on and people looking for this over about a decade, there was not a single new regulator, positive regulator found, and now in one CRISPR screen, we have 9. Okay, so this really was a testimony to us on the power of the full knockout in full genome CRISPR screening, and we had similar results with the negative. So Ching Li, the graduate student, he pulled this together, had his manuscript written. We sent it off to Nature Genetics, and one of the reviewers came back and said, they all liked it, but one of the reviewers said, you know, there's this new library out from the Broad, and it's called the Brunello library, and it's supposed to be better than the Gecko2. We'd like you to run your screen again using the Brunello library. Now, you're a graduate student, right? And this is the only thing standing between you and your PhD. So what do? Do you fight it? No, you go back into the lab and you spend 2 months doing the screen. So that's exactly what Ching Li did. There's a happy ending to this. So he does the screen, and now we look at the experimental false discovery rate of the Brunello versus the Gecko false discovery rate. And the correspondence is beautiful between the two libraries. And there's only a couple of genes that come up using the new Brunello that we didn't see in the Gecko. And it turns out the most important gene came up. So with all of the words that we said, when that review came back about running the screen again, the reviewer actually did us a favor. And that particular target made it into the title of the paper that we published in Nature Genetics. So it really is the main barrier to getting endoderm is the brakes that Junk June puts on from the step going from IPS into definitive endoderm. So, you know, we were really pleased with the results of this screen. We found so many new positive and negative modulators. It helped us understand organogenesis and then bring forward possibilities for cellular therapies. We had these strong phenotypes and really validated the ability, which we continue to use today, the Brunello library, as our full genome pooled library. So with that, that's the end of the third story. I have one more, and I think I have time for it. And it's a little bit different direction. So in the CME, I said I was going to talk about small molecule screening. So everything up until now has been functional genomics with RNAi and with CRISPR-Cas9. Now I'm going to mix in a little bit of chemogenomics, so using small molecules to get some very desirable results. So as Molly said in my introduction, I was with Roche Pharmaceuticals for a while. I was with them in 2004 when we published this paper in Science magazine. At that time, we were interested in developing therapeutics to break up the MDM2 P53 interaction and allow P53 to be active. So we ran a full genome small molecule screen. We had about 500,000 molecules in the collection at that time. We kept the most interesting hits to ourselves, and the ones that were also potent but much less patentable, we published in a science paper that showed that you could get this molecule here, which we called Nutlin because it was discovered in Nutley, New Jersey, just outside of New York City at the Roche site, to break up that interaction between MDM2 P53. And our thinking at the time was we don't know everything about this regulation, so why don't we give this tool molecule out there to all the academic sites working on P53, let them publish, and we'll learn as we go and continue to try and develop the best of the analogs. And there's close to 1,000 PubMed citations for Nutlin these days. So it's I think one of the best tool molecules really ever published by industry. And if you didn't know, there's about 110 high throughput small molecule screening sites, academic, in the US. I was an advisor to the MLSCN, the screening centers that would develop in academia under Zuhoni when he was the head of the NIH. And so about 400 different tool molecules, new tool molecules were put out there in that 10-year period. That became really good probes and increased our knowledge of cell biology. So this particular project was a collaboration that we did with Hebrew University. So like Don Way, Hebrew University was interested in developing iPS models towards therapies. But one of the problems with therapies, and this was also published news, that if you inject iPS cells, differentiated cells, whether it's dopamine expressing neurons or pancreatic cells, if you leave a few of the pluripotent cells over, you have the possibility of a teratoma developing. So you really want to wipe out completely any undifferentiated cell before the differentiated cells get into the patient. So our goal at that time was to create ultra pure populations of differentiated cells. If we had a small molecule that could wipe out the undifferentiated and keep the other ones going, okay? So safer ES and IPS treatments. And then at the same time, because there are so many similarities between ES, IPS cells and cancer stem cells, learn something about cancer biology. So that was the goal. So I worked with a graduate student at that time. Again, this is the fourth module and another graduate student driven piece of research. So these are the CSES cells that we use. These are human embryonic stem cells. We grew them on Matrigel with M-teaser media. This is low mag, this is high mag. These are beautiful, morphology colonies that are growing up, very easily stained. This is the graduate student, Yuri Ben David, who has his own lab now in Tel Aviv University. But the first thing to set up the screen is we needed killers, right? We needed positive controls. So this is Yuri weighing out the most toxic of our compounds that we could use as positive controls. And the one that we settled on was amsicrin hydrochloride, and that's shown down here. It's a DNA topo inhibitor, and it has both proliferation and non-proliferation based killing effect. So we thought, well, that's great because we don't know at what stage the cells are going to be at during the screen, so we need a complete killer. And then the one right next to it, amsicrin hydrochloride, that became our EC50 in all of the experiments that we're doing, because we wanted to know, is our screen drifting up or is it drifting down? So, you know, for the grad students and in the group, I'm going to show you exactly how we did this. So this is 2 screens a week, Monday to Wednesday, Wednesday to Friday. So 2 runs. We're doing about 7,000 to 10,000 compounds a day in 384 well format. So this is Yuri doing that cell culture. And then the plates were coated with Matrigel. After the coating, we would flick away the excess. We would harvest them with Accutase, count them on a Cedex, and then we dispense them, which you see here, a big jug of dispersed cells into, using this combi, into every row of a 380 well plate equally. And then we would add our positive negative controls with the rapid stack. So these are the 50 ml conicals that have positive EC90, EC50, as well as the negative controls with the tubing that are going to be added in the first 2 columns. And then, of course, to do the experiment, we have to take the experimental plate, which has the stem cells, with our compound plates. These are clear bottom for imaging. These are solid bottom plates that have the chemicals diluted to the right amount. And then you use the plate map and the barcode to match that onto the experimental plate that you're going to be using. So we would add the readout we decided was cell titer glow from Promega. We would add those per plate using the Biomech FX. And then we would stack these onto the Envision luminometer. So the readout was luminescence. We would load this up at the end of the day, go home, and next morning we had all of our results from the plate reader rastering across the plate. So here's an example of one of the 384 well plates. So these are the blanks up here in this corner. These are the EC50s, and these are the 100% amsicrin. So these are the stem cell killers. And you can see across the plate, we have a number of moderately or very active compounds showing up that we would later, do a follow-up on. And we're very pleased with the Z prime. If you don't know what the Z prime is, it's a term that Zhang published back in 1999. So it's taking basically the standard deviation of the positive and negative controls over the mean of the positive and negative controls, 1 minus that. The closer your number comes to 1, the better the screening metric that you have. So anything over 0.5 is considered a very good screen. So this is the primary screening results. This is the majority of the 52,000 compounds here, but you see a number of them are higher here. Some of them are dropping down. If you looked at the distribution, it was a beautiful, Gaussian bell curve. We chose an arbitrary cutoff here of about 50%, so we could look at all of the killing compounds up in here. Remember, we don't know yet if they're specific for stem cells. They're just killers. This is the Z prime. Every color code is a different run here. So really in 6 runs, we got through 52,000 compounds with a really high Z score. Not a single plate, dropped below the 0.5. So we didn't have to repeat anything. So now in confirmation, we took 3 human embryonic stem cell lines, the original and 2 other ones, to make sure it wasn't a stem cell specific effect that we were seeing. And then on top of that, we said, well, let's get away from stem cells and really see in HeLa, would it only kill the stem cells and not the HeLa cells? And then we said, well, let's make sure we can trust our readout. So although we had a luminescence readout, maybe, something was interfering with that. Let's take a fluorescent readout and compare those side to side. So that was our follow-up assay. This is looking at the same embryonic stem cell on 2 different days, very high correlation. We were pleased with that. Then this is looking at 2 different stem cell lines, H9 and CSES2, very high correlation. So it's not embryonic stem cell specific. And then here's where everything fell apart, the HeLa, right? So now this is a good indication. If it's not killing HeLa, but it's killing our stem cell, we have a chance of finding the kind of compounds we're looking for. So our counter screen now became taking those hits and going further and further away from iPS cells. So going down into ecto, endo, and mesoderm, and then further differentiating into things like hepatocytes, cardiomyocytes, fibroblasts, and then hopefully our killing effect would disappear. So our collaborator named these compounds pluricins, which stands for pluripotent stem cell inhibitors. So this is our red-green reporter here. These are the red cells that are undifferentiated. And you see that the fluorescein wipes them out. And in taking these very same cells, we differentiate them. And in fact, there is no killing going on at all. So here is the same compound, ES, IPS cells, kills effectively. ES or IPS differentiated, nothing. Fibroblasts, nothing. Now you can see really the gradation as we go further away from embryonic stem cells into differentiated. You start to lose all of that killing capacity. At best, you might get a little bit of mild toxicity down here, but by and large, it's gone. So the whole screen of 52,000 looks like this. We started with 52. We had about 2,000 primary hits, 700 confirmed hits, and then through further validation, about 15 absolutely defined fluorescents. So what do they look like? Here's just a snapshot of a number of the chemical series. Some of them are related. There's no doubt about that. But there's others that are very unique structures. And so in looking at the literature, what do these structures mean? And it always boiled down to inhibitors of steryl-CoA desaturase, or SCD1 inhibitors. It's a key enzyme in monounsaturated fatty acid biosynthesis. It's an ER membrane protein. And there was some literature to suggest that ER stress and apoptosis in cancer line was linked to SCD1 inhibitors, but it had never been studied before in either embryonic or IPS cells. So we looked a little bit deeper and the story kept building around SCD1 and cancer cells. Here's some of the publications that we came across, tying that in. So then now to finish this up for publication, we had to do the big experiment. So this next slide is a little complicated, so I'll walk you around it. So here with the knockdown or the biochemical treatment of SCD1, you could see that we get a very good knockdown. It's involved in ER stress pathways. It's involved in particularly late apoptosis. That protein synthesis is not really involved. And I think maybe the biggest proof is right up here, which is when you add back OA, you can take oleate and add it back to the same IPS and ES cells that are going to be killed by the fluorescein, and you can compete it off. So we felt very good that in fact the SCD1 was going to be the target. But we had 15 hits, and some of them looked very different. And in fact, there were others that you couldn't compete off. So there definitely was another mechanism going on that we had to study afterwards. And then getting back to the original premise, well, now other people picked up on the work after we published it, and here's some work where they're using ES cells on, these are on mice. So what they're doing is injecting them into mice with either fluorescein treated or non-fluorescein treated. The ones that are non-fluorescein treated produced a teratoma. The ones that were fluorescein pre-treated before injecting into the animal had 0% of working with teratoma. And you could see those numbers over here. And even when we did a one-to-one mixture of a different cell type, it wasn't enough to dilute it. So the fluorescein always killed the undifferentiated stem cell. And then people started doing now, so this is a model of cardiac infarct. And so the investigator who wrote this paper used the fluorescein pretreatment and injected it. And in fact, with that, there was a good cardiac healing of the event that had happened, and there was no teratoma, no other aberrant physiology that was growing in. So we felt very good that we had met that criteria. Specific stem cell killer was usable for pre-treating IPS or ES cells that would be used therapeutically. But what about the other side, right? Because we're all cancer biologists. What did these compounds tell us? And there are hallmarks, right? Self-renewal, rapid proliferation, telomerase activity, gene expression patterns, and epigenetics. So there are definitely a number of parallels, including genomic instability, that IPS, ES cells have, along with various isolated cancer stem cells. So we did the experiment, and you could see it here that in the normal cells versus these cancer stem cells, that you get a very good killing with these fluorescins if you look at it. So this is fluorescin 1, and I think these are glioma-based cancer stem cells that were looked at. So definitely a possibility of creating a therapeutic that would be used either singly or in combination to wipe out the cells that might, and you all know this, that when we treat the tumors with existing therapies, that there's always that subpopulation thought to be stem-like in its appearance and function that are going to come back and rage even worse than the primary tumor did. So one can envision a type of therapy where you're going in with the modality that's accepted, and then a different dose, a different modality to attack the few remaining cells that might morph into the resistant cells. So with that, I'm just about on time. So I was told I have to put this up, which is so you can answer a brief survey saying how good the pizza was, whether this was a waste of time or not. And before you fill out that survey, remember, go Buffs. All right, so thanks very much.
Hey, beautiful work. I'm curious about the second project on the oncolytic viruses, on SVV. Are there normal cells that produce the TEM8 receptor, and how is it that cancer cells end up producing this receptor?
I mean, I'm not an expert on TEM biology. I've just been using it as a tracker. Now there's this whole another group that wants to use it as a biomarker. So it's one, as you know, it's rare for a cancer to put a red flag on its surface. So this is one of the rare ones. It's not in every neuroendocrine. It's not in every pediatric cancer, but it's in a high percentage of it. So, you know, much like, you know, HER2 is a biomarker that we use for Herceptin. That's in roughly, what, 25 to 30% of all breast cancers. Everybody else should be excluded from that trial. So that's what we're trying to do with that. In terms of how it helps the cancer grow, what selective advantage, because obviously it's making it, I can't speak to that. I'm not an expert in that. Yeah. Oh, sorry, right. Yeah, I should, sorry. Sorry, they can't remote, sorry.
That was four really interesting vignettes, and thanks so much. I have a question about two of them. One, the TIM8 story. I thought it was interesting with the Seneca Valley virus, using that as a receptor. And I was wondering if you could tell anything from the screen, like what was co-regulated that might be what's controlling the expression of the TIM8. You didn't catch it? The TIM8 and the Seneca Valley virus story. Yeah. So from the screen, could you tell anything that was sort of also coming up that was a potential determinant or regulator of whether they were expressing TIM8 or not?
Yeah. So, I mean, in truth, we were thrilled to find the receptor, the TM8, and the interferon involvement. So that project right now is waiting for investment. So it's gone through 3 different owners. I don't know if I did it, but it was Neotropics, Perceiva, and now it's Seneca Therapeutics. And Seneca Therapeutics only has about 3 or 4 employees, so they're really waiting. And I think their next big break is going to be with a company that wants to make a cold tumor hot and use, you know, just a very small, brief dose of that to do it and allow a CAR T or a natural killer cell to get in.
Yeah, but like there's some neuroendocrine therapy or neuroendocrine cancers, like say prostate cancer, when it becomes really aggressive, it turns into like a neuroendocrine that might not be expressing the TIM8, but if you could induce it to express, then you can.
Right.
But okay, and my other question was about why the Brunello virus or CRISPR screen works better than the gecko, and why you would come up with this
Yeah, so,
particular target that was just totally missed if they're both supposed to be comprehensive?
yeah, so they both work well. I mean, the gecko worked in the Seneca Valley, and there was no reason to go back and do it in Brunello. But for the pancreatic differentiation screen, we had great correspondence, and a lot of times you'll look side by side, and the guides by the old rules are identical in gecko and Brunello, but in this case for the June, they were all different. So the new algorithm had developed much better on-target guides than we had for the gecko. So yeah, gecko is, it's useful, but Brunello's better. And I'll tell you an anecdotal story. I told it at dinner last night to Molly, which is, as a core lab, you can't always build it and they will come. When the first full genome CRISPR library came out, Gecko 1, we bought it from Adgene, we amplified it, we made maxi preps, we QC'd it to make sure it wasn't underrepresented. And like a month later, the Broad came out with the Gecko 2. So in my freezer in Manhattan is the full Gecko 1, never been used. Okay, so we worried that this was going to happen again and again. Luckily, we had at least I'd say a 3 or 4 year run with Gecko 2, and now Brunello since 2020. So it's having a 5-year run as the best designed library. But we were worried, that it was going to go A, B, C, D, right down the line with newer versions, but it hasn't happened, luckily. So being first in is not always an advantage. Yeah.
That was great. I had a question about moving from in vitro screens into in vivo screens. So there's always been a lot of people using Brunello in these large-scale in vitro systems, but obviously you cannot achieve the coverage you would need in vivo. Do you know of anyone that really validates their hits using an in vivo, maybe targeted approach, maybe a targeted library? And what is the threshold of guide coverage that you've been able to achieve in vivo?
Yeah, so your question's about targeted libraries, right, versus full genome?
And going and doing the screen in vivo, not in a dish anymore.
Right.
Yep.
Yeah, so I have data on that. We have screens where we've made, you just need to come to us with a gene list. So it could be a known family, it could be all the kinases, all the phosphatases, all the genes involved in the epigenome, DNA damage response. We have all those libraries. Or you might have a gene ontology list and you come to me with your custom list. We did a project with a NYU investigator where they're really interested in all the calcium regulated channels in the heart, crack channels. And so we made a custom library for them. We told them we wanted to resell it to other investigators. They said, you can do that, but not until we publish the paper. So we actually had to wait 4 years, and then they published that paper, and now that's available in our war chest. But there's a lot of advantages to doing focused libraries, right? So you're not working with 150 plates per screen. Maybe you're working with 30 plates or 40 plates. So the scale of economy, and that also allows you to go to a higher end. So, one mistake we see quite often is people do an N of 1 on this. And as our bioinformatics expert says, 1 is none, 2 is 1, 3 is 2. So you always want to have at least 3 replicates, either scientific or technical, to be able to get the right answer out of it. But yeah, there's great power in focused libraries. I just didn't have a chance to show it today. Yeah.
I'll make this quick. So, the general word on the street's always been that, SI or shRNA screens allow you to do sort of what Scott Lowe at your place calls an epiallelic series. You get variable knockdown, but that could actually be an advantage because sometimes a complete knockdown may just be a complete lethal and not give you what you're looking for, versus CRISPR, which is kind of all or nothing. Has that kind of held up with your experience? Because I've never seen any actual empirical data to show that that is the case.
Yeah, great question. So, as I showed in the first screen, there are cases where SI and SH works great. Okay, and you know, people didn't stop developing. Scott Lowe kept developing SH technologies. We use MIR-E now, high efficiency SH, which in a lot of cases gives you just as much knockdown as you're getting knockout. And let's just talk about knockout for a second. So a lot of those indels are in frame. So they're not really 100% knockout. If you looked at the Seneca Valley virus, when we went into other cancer cell lines, there was a little bit of residual activity. So that is due, we believe, to an in-frame mutation. So even an absolute knockout in many cases is not an absolute knockout. Plus, because the RNA technology over the last 25 years, since Fire and Mello won the Nobel Prize, has continued to develop. So people haven't rested on their laurels. So now I think the gap between SH and CRISPR is much more narrow. And so a lot of times investigators will come to me for a letter of support for their grant, and I'll write it up and they'll say, oh, we can't put RNAi here. You know, even if it's a phenotypic readout, I say, okay, then let's lead with CRISPR, and you can put the RNAi as your pitfall, as your fallback position. But sometimes when they get the money, we wind up doing the fallback position first because it makes more sense to use a RNAi-based technology instead of CRISPR.
Great, thank you.
Question, though. Yeah. Okay. Thank you. Oh, good, good. So almost nothing here you're gonna see next week. Then you realize that's because I'm gonna take this next week. On how to work with the reagents, the tools, the financials, all that. I'd like to really make, because I think that's what that one wants. Yeah. That will be very brief.